So my name is Benjamin Wallace, and I'm a member of the Mullally Lab. And I have the great honor to talk today a little bit about biomarkers in myeloproliferative neoplasms. And due to the fact that I only have a maximum of 15 minutes, it's not really a talk. It's more like a brainstorming session. So when you have any questions, just interrupt me at any time. So the definition of biomarkers is not clear. So what is a biomarker? So the term has two parts. So a biological measurement, and the second part is the marker. So something we like to associate the measurement with. So biomarkers can be used to indicate an increased risk of disease development. A biomarker can indicate the diagnosis itself. It can be used for risk stratification, for disease monitoring, or for treatment monitoring. And I very much have to excuse that I start very, very basic. So we know that we have different flavors or subtypes of MPN disease, namely polycythemia vera, essential thrombocythemia, and primary myelofibrosis. And these diseases are very different. So PV is characterized, and I know that everybody of you are aware of that, it's characterized by more red blood cells. ET is characterized by more platelets, and PMF is characterized by more stroma in the bone marrow. But even though these diseases are very different, we find same driver mutations in all of these subtypes of MPN, namely mutations in JAK2, mutations in MPL, and mutations in CALR. And all of these subtypes of MPN disease are associated with these driver mutations. And what is quite striking, these mutations are quite recently discovered. So it's not so long ago that we knew about these driver mutations. So the first one that was discovered was the JAK2 mutation, the most common mutation in MPN. And the first one that was discovered, namely V617F. And this is kind of our first biomarker. So something that we know that is well established in most cancer centers of the world. And does it make sense to really measure these driver mutations? And the answer is yes, of course, it makes sense to measure driver mutations. And I just put out two examples. The first example is for JAK2 mutations, the variant allele frequency, so the allelic burden of a JAK2 mutation. And there are data showing that higher variant allele frequency is highly associated with an increased risk of thrombosis. So the allelic burden is a biomarker that can indicate certain complications. And what is now increasingly important, for example, is detecting CALR mutations. Why is that important? So there are newer trials, upcoming trials that are targeting CALR mutant cells. So there are now trials focusing on or by using monoclonal antibodies to target CALR mutant cells. There are some trials focusing that use CAR T cells, and there are also trials that try to vaccinate against CALR mutations. But before the driver mutations itself, what is quite strikingly is that there are now several papers in the literature that indicate that there are certain germline variants that predispose to MPN development. And I was very much surprised to see that the heritable component of MPN is very high compared to other cancers. So it seems like a lot of MPN diseases are driven by certain germline risk genes. And some of these genes are, for example, TERT or POT1. These are genes that are important for telomere maintenance, leading to sustained telomere maintenance in hematopoietic stem cells, thereby driving mutant stem cell expansion, or variants in genes that are important for HSC self-renewal, like TET2, or genes that are important for stem cell self-renewal and DNA repair, like CHEK2. And there's also a group from Aachen that published that even well-known cancer risk genes like BRCA that is well known for an increased risk in for breast cancer, that even germline variants in this gene can lead to familiar MPN disease. And I'm pretty sure every one of you is aware of clonal hematopoiesis. And I'm mentioning this now because this is one of our main focuses in the lab right now. So what is clonal hematopoiesis? So we know now for several years that there are some healthy individuals in the general population that have that harbor some of these MPN driver mutations, for example, JAK2, but they don't have MPN disease. So, and some of these people develop over time MPN and some don't. And we completely don't understand which of these people develop MPN and which don't. So, and that's kind of one of our main angles, finding factors that promote JAK2 mutant clones or impair JAK2 mutant cell expansion. And it's very interesting to see there's an amazing paper from Egarin et al. Showing that the acquisition of these mutations, for example, the JAK2 mutation, takes place in many patients very early in life. So in some patients in utero and in some patients in the early childhood. And then these clones stay there for a certain period of time. And we know that the variant allele frequency can change, it can increase and decrease over time in these individuals without having MPN disease. But at some point, there's a factor or there's something's taking place that we don't know of, that these patients suddenly or over a certain period of time develop MPN disease. And we are trying to find out what are these factors, and do we have maybe potential biomarkers indicating that some people develop the disease and some don't. So I talked now about germline variants and certain risk of MPN development and clonal hematopoiesis. But what about biomarkers when you already have an MPN disease? And the left figure that you see here now is a poster that we presented during ASH 2023 in San Diego, and it's showing MPN patients dependent on certain risk mutations. So the yellow graph, you see PV and ET patients. In green, the green graph are patients only with bone marrow fibrosis. And then the blue graph indicates TP53 single hit patients, and the orange graph are TP53 multi-hit patients. And the main finding of this small project that we are currently expanding is that fibrosis itself is a risk factor. So that's the green graph that you see here right now. And that the real patients at high risk are patients with a TP53 double hit mutation here in this orange graph. And so that these are kind of biomarkers, fibrosis itself and TP53 double hit indicating a decreased overall survival. And same findings are also published for other co-mutations, for example, A601, EZH2, EDH1, EDH2, or SRSF2. Independent of these co-mutations that I mentioned before, there are even more basic measurements that are more basic clinical parameters or biomarkers that are associated with prognosis, so that we kind of close to daily measure in clinic. So one example would be the lactate dehydrogenase, the C-reactive protein, or blood counts itself. And I think that's something that we close to always do in clinic. So for the lactate dehydrogenase, it was shown that higher values are associated with more complications in the sense of and disease progression to secondary AML, and also in decreased overall survival and leukemia-free survival. The C-reactive protein kind of indicates that MPN is a really pro-inflammatory disease with a lot of pro-inflammatory cytokines, and higher values are associated with more thrombosis and decreased overall survival. And even blood counts itself can be used to monitor MPN disease over time in some subtypes, but also it was shown that increased leukocytosis is associated with increased risk of thrombosis, and that's also something we focus at in the lab, looking at certain leukocyte subtypes, how these cells can induce thrombosis. For example, in the case of monocytes, how tissue factor expression on monocytes may lead to increased thrombotic events. So next slide. So as I mentioned now, monocytes itself seems to be a biomarker for an increased risk. So what you see here now is a very beautiful abstract from ASH 2016. So you see here now PMF patient categorized in different DIPS risk categories. And you see in green, so I adapted the figure a little bit. In green, you see PMF patients with a high DIPS score without monocytosis. And in red, you see PMF patient with a high risk score with monocytosis. And you see that the presence of monocytosis alone is a biomarker that indicates an decreased overall survival in PMF patients. The next slide. And some labs focused on that in more detail. So there is a really good paper showing that especially pro-inflammatory cytokines drive disease progression in more specific SLAMF7 expressing monocytes, so that these monocytes are able to promote bone marrow fibrosis over time, what means that these cell subtypes may be a potential target that we can use for future therapies. And another great paper published by the Schneider-Kramer group showed that another pro-inflammatory cytokine, so not the C-reactive protein that I showed before, in this case now CXCL4, that's also a cytokine that can be expressed in the bone marrow, and that higher levels are associated with increased risk of fibrosis, showing that maybe this cytokine could be a potential biomarker that indicates an increased risk of fibrosis. Next slide. Yeah, and there are also a few papers indicating on newer biomarkers, so comparable to projects focusing on solid cancers, where labs try to early detect solid cancer by measuring cell-free DNA in the bloodstream. There are now also groups trying to measure cell-free DNA in MPN patients to early detect, for example, thrombotic events or other complications. And there are also other labs trying to do the same, so trying to find association between complications and extracellular vesicles, microparticles in the bloodstream, for example. So as microparticles, we understand other cell components and or protein components from a cell, or circulating endothelial cells, and trying to associate the measurements of these parameters with complications. But it's really at the beginning. There's already a review article about that. You see that now here on the screen. But how valid these measurements are, and if we can use them for future risk stratification MPN patients, is quite unknown at this point. Yeah, next slide. And to kind of summarize it, and I know that here right now currently a lot of great researcher in the meeting, so and I hope that I can motivate all of you and to emphasize that we have already good biomarkers, but we have too less, so we need more and better biomarkers, and we need especially biomarkers that are good in monitor the treatment that we are currently doing in our patients, but also to select treatments so that we can go more into a personalized medicine, so that we can decide, for example, what patient we have to treat with interferon, or which patient we have to give early stem cell transplantation or which patient needs another treatment. And because I'm right now put a lot of stuff in the presentation what we are interested in the lab. So we are also very much interested in finding biomarkers that especially predict adverse events due to the fact that MPN disease is a disease that's more or less chronic and most people live for decades, except when these patients develop certain complications such as thrombotic events and or the development of secondary leukemia. So there's a high need of finding biomarkers that show which patients are at high risk of developing thrombosis and or leukemia. Yeah, thank you very much for your attention, and I hope that you have a lot of questions.